What's up, TUMNAD lectures? In this video, we are looking at what we call the bile synthesis. If you find it helpful at the end of it all, please make sure you like the video, comment down in the comment section, and then subscribe. Now, in the previous video, we have been looking about the bile flow in the biliary system. And we talked that the bile is going to be having a lot of components. It's going to be having what you call the bilirubin, going to be having what you call the bile acids, going to be having the xenobiotic, the phospholipids, the cholesterol, the immunoglobulin A, and the electrolytes, the water 95%. We talked about all those kind of the components that make what we call bile. Now, in today's video, we're going to be looking how do we synthesize some of those components of the bile. Mainly, we are going to be focusing on what we call bilirubin and even also what we call the bile acids. But in today's video, we are going to be mainly focusing on how we are going to be making what we call bilirubin. Now, you need to first understand, where do we get bilirubin? Now, bilirubin is going to be coming as a result of breakdown of what we call red blood cell. Now, we need to understand, where do we find the breakdown of red blood cell? There are very many body structures that are going to be having what we call breakdown of red blood cell, even also in the blood vessels. But particularly, we are going to be looking at the breakdown of red blood cells in what we call the spleen. Okay, because we have worn out red blood cells. These red blood cells that have clocked their 120 days, they are now starting now to meander around. So now they need to be broken down because their flexibility is now taken over. They cannot squeeze through what you call these capillaries. Okay, so now this is going to be now the spleen. Okay, and of course it's going to be now our blood supply to the spleen from this blood vessel, which is a branch of the celiac trunk. You know how this artery is called? This one here is going to be what we call the splenic artery. It's going to be supplying oxygenated blood, what we call the spleen. Now, in the spleen, you're going to be having a type of the capillaries which we call the sinusoid. So now this one here is going to be what we call the sinusoids. Okay, and of course the sinusoids are going to be lined with what we call the endothelial cells. Now, we're going to be having the worn out red blood cell. These red blood cells which are now becoming old. It's their time to go away. They need to be broken down because they cannot squeeze through the sinusoid capillary. And now, within these capillaries, we are going to be having this the specific white blood cell. Super very important. This white blood cell here, this one here is what we call the macrophage. Okay, macrophage. So now we are going to be having tons and tons of macrophages that are going to be residing within what we call the spleen. Okay, so now these macrophages are going to now start breaking down these worn out red blood cells. Okay, now we are going to be now seeing now these red blood cells getting phagocytosed into what we call the macrophages. So now we are now going to be assuming that this red blood cell is now taken over by this the macrophage. So now you're going to be zooming out to the macrophage, and we see exactly how the macrophage deals with this red blood cell. Okay. Now, when this macrophage takes in what we call the red blood cell, it is going to be now breaking that red blood cell into different fragments. In this case, I'm going to be mainly focusing on what we call the hemoglobin. Okay, I'm going to be mainly focusing on what we call hemoglobin. Okay, and even also don't forget there are other structures, the cell membrane, and even other of components as a result of the breakdown of red blood cell. But I'm mainly focusing on what we call hemoglobin. Okay. So now, when this hemoglobin is now going to be further broken down, it is going to be broken down into two components. Okay, it's going to be broken down into what we call heme. Okay, it's going to be broken down into what we call heme, and even also what we call the globulin. Okay, what we call the globulin. Now, the globulin is a long chain of amino acids. So now it can even get further broken down. It can get further broken down into individual amino acids. And of course, these amino acids can get used up by different body structures, like even also the liver, and even also some other structures that can need to use these the amino acids. Now, my main focus is going to be dealing with what we call the heme. Now, this heme, under the influence of an enzyme called heme oxygenase, it is going to be broken down into another kind of the molecule which you call biliverdin. Okay, so now this kind of the heme is going to be broken down into another molecule which we call biliverdin. Okay, what we call the biliverdin. Okay, now we have said that this process here is going to be aided by an enzyme called heme, which we call heme oxygenase. Okay, heme oxygenase is the one which is catalyzing the process of breakdown of heme into biliverdin. And of course, also we are going to be expelling what we call the iron. Okay, so now in the macrophage we have what we call an iron sac. We have a protein sac that's going to be storing what we call the iron. This kind of the sac is what we call the apoferritin. Okay, so now this iron is going to be stored into that kind of the sac which we are calling the apoferritin in the form of what we call ferritin. Okay, in the form of what we call ferritin. Okay, in the form of what we call ferritin. So now that's going to be now the storage form of what we call iron in what we call the macrophages. Okay, but also the liver also makes another kind of the transporter of iron which we call the transferrin. Okay, the liver also makes this kind of transporter which we call the transferrin. So now this transferrin can also bind iron and then transport to different areas where it's going to be used. Now, for example, it's going to be taken to the bone marrow, and of course in the bone marrow it can be used in the production of what we call the heme, because the heme is going to be containing what we call the iron. So now the transferrin can transfer this kind of the iron to other areas where this kind of the iron is so much important for use. Okay, so now that's for the case of iron. So now we are moving with what we call biliverdin. So now this biliverdin is going to be a green substance. Okay, it's going to be appearing green. Okay, so now this biliverdin is further reduced into another kind of a molecule. Okay, it is further reduced into another kind of the molecule. You know how this molecule is called? It is reduced into what we call bilirubin. Okay, it is reduced into what we call bilirubin, and this kind of the process converting biliverdin into bilirubin is aided by an enzyme. This enzyme is called biliverdin reductase. Okay, this enzyme is called biliverdin reductase. Okay, biliverdin reductase, converting biliverdin into what we call bilirubin. So now this bilirubin which has been formed is going to be lipid soluble, but it is insoluble in water, meaning that it is hydrophobic. Okay, it is unconjugated, it is indirect. There are so many terms you can use on this one here. So now this kind of bilirubin is going to be unconjugated. Okay, it's going to be unconjugated. You can even call it going to be indirect. Okay, it is going to be indirect. So now, for that reason, this kind of the bilirubin, the fact that it is unconjugated, it is not going to be water friendly. It is hydrophobic. We need to transport it to another structure where it is going to be getting conjugated such that it becomes friendly with the water, so that it can even be excreted in form of bile and gets stored into what we call the gallbladder. So now the question is, who transports this kind of the unconjugated bilirubin to the place of what we call the conjugation? Remember, we are going to be carrying this kind of the unconjugated bilirubin to what we call the liver, because the liver is going to be aiding in the conjugation process. Now, what happens that the fact that this kind of unconjugated bilirubin is lipid soluble, it can just diffuse out of what we call the macrophage. So now you're going to be seeing now this kind of the bilirubin. You're going to be seeing this kind of the unconjugated bilirubin coming off from what we call the macrophage. So now, because it's going to be insoluble in water, it needs to be transported from the spleen and gets taken to the liver for the process of conjugation. So now, the liver makes this kind of the plasma protein that aids in the transport of unconjugated bilirubin. You know how this kind of plasma protein, how it's called? This kind of the plasma protein here is what we call albumin. Okay, it's what we call albumin. So now albumin is going to be moving through the spleen, and then it will come within the spleen and binds the unconjugated bilirubin. And then after binding the unconjugated bilirubin, it will take through this blood vessel. So now what unconjugated bilirubin is going to be taken over by this spleen. You know this spleen, how it is called? This is going to be now our albumin. It is binding the unconjugated bilirubin and it is transferring to the liver for the process of conjugation. So now it is passing through this blood vessel. You know how this blood vessel is called? This blood vessel here is what we call the splenic, vein. So now this splenic vein is now going to be coming and joining this big vein here that enters the liver, which we call the hepatic portal vein. Okay. So now our the bilirubin now is going to get transported, to the liver. So now our unconjugated bilirubin has now entered to the liver. So now in the liver, we are going to be having what we call the hepatocytes. And of course, the hepatocytes are going to be what we call the liver cells. Okay. So now here we have just zoomed only on one of these liver cells, which we are calling the hepatocyte. It is going to be having what we call the basolateral side, which is going to be facing the sinusoid, and even also the apical side, which is going to be facing what we call the biocanalicular. Now, we are going to be now seeing this kind of unconjugated bilirubin. Now it has now entered inside what we call the hepatocytes. Now, what happened is that in the liver, we have an enzyme called uridine glucuronide transferase. It is that enzyme the liver uses to conjugate what we call the unconjugated bilirubin. So now, this kind of an enzyme is going to be aiding in the reaction between unconjugated bilirubin with what we call glucuronic acid. Okay, with what we call glucuronic acid. So now, unconjugated bilirubin, when it reacts with what we call glucuronic acid, under the influence of an enzyme called uridine glucuronide transferase, our bilirubin now becomes fully conjugated. So now you're going to be having now what we call conjugated. You're going to be having what we call conjugated bilirubin. Okay. So now, after having the conjugated bilirubin, it has to exit what we call the hepatocytes. We have a specific transporter of conjugated bilirubin on the apical side of what we call the hepatocytes. You know how this transporter is called? This transporter here of conjugated bilirubin is what we call multi-drug resistance associated peptide 2. Okay, so now this is the classic transporter that allows conjugated bilirubin to leave what we call the liver on the apical side. So now our conjugated bilirubin is now going to be flowing into this the biocanalicular, and then it will move and get stored into what we call the gallbladder. Okay, because the moment it becomes conjugated, it's now water soluble. It can get excreted and get stored into what we call the gallbladder. So now, if someone who has a problem with this transport we are calling multi-drug resistance associated peptide 2, it means conjugated bilirubin will not be able to move out of the hepatocytes. We shall be piling a lot of what we call conjugated bilirubin. So now, in that way, some little of the conjugated bilirubin will now start accessing what we call the general circulation. So someone may end up having what we call hyperbilirubinemia. Okay, having high levels of what we call bilirubin. Okay, so now, if anyone gets any kind of mutation of this the transporter, if any person gets a mutation of this transporter of conjugated bilirubin into the biocanalicular, it will lead into a condition called Dubin. It will lead into a condition called Dubin which we call Dubin-Johnson, which we call Dubin-Johnson syndrome. Okay, Dubin-Johnson syndrome. Okay, so now that's going to be a mutation of what we call multi-drug resistance associated peptide 2. Okay, so now our conjugated bilirubin now has been now stored in the gallbladder. Now from the gallbladder following a meal, so now all the components of what we call the bile are going to be now moving through this biliary tract system into what we call the intestines. Now when bilirubin, which is conjugated, reaches into the intestines, in the intestines we have what we call bacteria having tons and tons of different kind of the enzymes. Now this kind of the bilirubin, when it flows into the intestines, is going to be now broken down by what we call bacterial enzymes into another kind of the molecule, which we are calling urobilinogen. Okay, which we are calling urobilinogen. So now from conjugated bilirubin into urobilinogen by the influence of what we call the bacterial enzymes within the intestines. Okay, so now what happened is that some of the urobilinogen is going to be now retaken back. It's going to be now absorbed on the terminal area of what we call the ileum back into the liver. Okay, so now this kind of the absorption through what we call the superior mesenteric vein on what we call the terminal ileum is going to be absorbing this kind of the urobilinogen back into what we call the liver. So now this kind of the movement from the liver to the intestines and back to the liver, this is what we call enterohepatic. This is what we call enterohepatic circulation. Okay, this is what we call enterohepatic circulation. So now this kind of the urobilinogen that enters into the liver, it will eventually leave through what we call the hepatic vein into the general circulation, and then circulates what we call the heart, and then finally it will find itself in what we call the kidneys. Now in the kidneys, it's going to be broken down into this kind of this molecule, which we are calling the urobilin. Okay, the urobilin, and this urobilin gives the yellow color of urine. Okay, it gives the yellow color of urine. Okay, so now the yellow color of urine is as a result of what we call the breakdown of what we call urobilinogen into what we call urobilin. Okay, then some of the urobilinogen is going to be continuously moving within the intestines, and is going to be again further broken down into another kind of the molecule. It is going to be broken down further into another kind of the molecule, which we call the stacobilin. Now this stacobilin is one which gives the fecal matter the color as brown. Okay, so now our fecal matter is going to be appearing brown just because of what we call the stacobilin, due to final degradation of what we call urobilinogen by intestinal microorganisms into what we call stacobilin. Okay, so now that briefly covers the synthesis of what we call bilirubin, and how it is going to be moving through the intestines, and how it's going to be turning the urine into yellow, and even also the stool into what we call brown. Okay, so now TUMNAD, I have talked a lot of things, and if you think whatever I've talked is making sense, is adding to you, you're benefiting, please you can like the video, comment down in the comment section, and then subscribe. So now in the next video, we are going to be now focusing on what we call the synthesis of what we call the bile acids. Okay, thank you very much for watching. TUMNAD as always, until we meet next time.